Participants, and I think more people are joining in, so we'll just start. So, hello, good afternoon, everyone. Welcome to our monthly research seminar. My name is Salik. I work as a teaching and technical assistant at Sangath Bhopal. I will be your moderator for this session, and another moderator for this session will be Babita Negi, also from Sangath Bhopal Hub. The structure for this seminar we'll have a brief introduction of the speaker, following which our speaker will talk about for approximately 30 to 45 minutes, and then about half an hour of Q&A. So that is the structure. About the seminar, I'd like to say that you all know that years ago, we started with this idea of monthly research seminar with the intention of capacity building for the employees of Sangath. And this is another seminar in this series. We are live on YouTube, and this seminar is being recorded, and it will be shared and uploaded on the social media accounts of Sangath, and it shall remain in public domain. A couple of housekeeping announcements. I'll request everyone to kindly mute themselves. Make sure that you're mute. If you have any questions, a comment, you can use the chat box, and you can raise your hand and unmute yourself during the Q&A. All right, so with the housekeeping announcements done, let's come to our today's speaker. Starting her academic journey from St. Xavier's College in Mumbai, she went on to pursue a doctorate in neuroscience at Yale University in the United States of America. She later pursued postdoctorate research at Karolinska Institute in Sweden, and later at University of Oxford in the United Kingdom. She joined the Tata Institute of Fundamental Research in the year 2000 and is currently a tenured professor there. Her laboratory uses mouse and rats as model organisms to explore and answer questions of the neurobiology of emotions. She is currently an elected fellow of the Indian National Science Academy, that is INSA, and in the past has been a senior overseas research fellow of the Wellcome Trust. She is also a senior editor of the European Journal of Neuroscience. A prolific speaker, a scientific outreach enthusiast, and a firm believer in gender equality, Professor Dr. Vidita Vaidya stands tall as one of our country's leading neuroscientists. We at Sangath are very delighted to have you with us, Vidita. And with that, I request and invite you for the further proceedings of this seminar. So over to you, and thank you.
Thanks, Salik, for that very kind introduction. I'd like to thank Anant and Salik and Babita for organizing this seminar today. I'm really looking forward to interacting with people at the Sangath team. You do the real work. We get to sit in our laboratories and ask questions, but you are the ones who actually make straight out impact to human lives. And so the opportunity to interact with you is an honor for me. Okay, I'm going to share my slides, just confirm that you can actually see them before I get started. Salik, can you just confirm that you can actually see my slides?
Yes, we can see them.
Okay, yeah. All right. So I'm going to talk to you today about early adversity and early stress and how that is a common risk factor across the board for a diverse array of psychiatric disorders. I want to start with the caveat that a lot of my work will come from animal models, and there is always the risk that when you extrapolate data from animal models to humans, things are lost in translation. However, there are also commonalities, and it is the commonalities that I will stress upon, but do keep this caveat in mind when you're directly extrapolating the findings that we have from animal models. Just something to keep in mind as a concern. Okay, so let's start with something that is not a model organism, the human, the brain that we're perhaps most fascinated with because it is really studying our own nervous system in a sense. Here is a cartoon video actually from the HHMI Biointeractive website. We'll just give you a sense of the early stages of development and what it looks like. So what you start with is actually this tube-like structure, which is filled with the cerebrospinal fluid. And then unevenly around this, you will see proliferation of cells. And now zooming in on the ventricular surface, you can actually see that there are radial glial cells or stem cells, which send out a process towards the pile surface. And then there is this profound amount of cell division that takes place. These are neural stem cells that divide and give rise to all the cells that occupy your nervous system, in a sense. And here you see the structure actually getting filled up. And I want you to give you a sense that most of the cells are in place at birth. So really, during that 9 months of early development, you have profound cell division and a substantial amount of neurodevelopment already taking place, which is one of the reasons why when the baby comes out, you also realize that the head is the largest thing that you have to support the neck up, to really hold this nervous system up and support it eventually. And we are born fairly immature in terms of our ability to move, unlike many other species. But the nervous system really has put in a lot of effort and energy to put the structures into place. And this is a sort of a leapfrog model where the youngest cells tend to actually be on the outer surface and the older cells are closer to the ventricular surface. So this is how the nervous system develops and forms. And what I want to tell you is that it's remarkable how nervous systems, all vertebrate nervous systems, including mammalian nervous systems, use a fairly well-conserved genetic blueprint in which perhaps the details of the recipe may vary, but really the broad principles are fairly similar to put together the brain. What you see on the left-hand side is a rodent brain, rat or mouse. What you see on the right-hand side is human. And it's obvious that it's already quite different, and you can really see the difference between these two nervous systems. And yet there are commonalities, and it's the commonalities that I want to stress upon. And the commonalities I'm stressing upon here, the yellow, for example, is that part of your brain that controls motor control. So my ability to produce speech is being controlled by my motor cortices as we speak, as I literally speak. That's information that's coming from my motor cortex, then down to my larynx to actually drive movement. The somatosensory component, which is in red, is again in similar positions in both the rodent brain and in the human. And vision, we all process vision. So those of you who are looking at my sides are currently having your visual cortex, primary V1 in your occipital lobe getting activated as you stare upon these slides, right? So why am I showing you this image? What I want to tell you is that there's a genetic recipe book which is placing these structures in the brain so that everyone who's processing vision, all mammals, are processing it in their occipital lobe, right? Suddenly, you will not have this blue structure move to the front, which is where the prefrontal or executive function of your brain is done. It is done like a recipe book so that every human that's walking the planet has their V1 in their occipital lobe at the back of the brain. And this is reliably done. And yet we are so different from each other in our detail. We are so different from each other in many, many things that we do. So where does the variation arise if there is this genetic recipe book that is used to generate this brain structure? Well, you remember we all start with a single cell. And then from the single cell, you're going to generate complexity. And at the level of the brain, you're going to generate this incredible complexity of 100 billion neurons, which eventually produce function by the manner in which they talk to each other. So this is to give you a sense that over time, and here's a newborn, over time, you're also adding a fair bit of stuff in place. I mean, especially in the cortex, there's a fair bit of change that's taking place. To give you a sense that it's not all done at development. Lots of things are already in place, but the connections, the pruning, the organization still yet to come. And this is where experience comes in. So in a sense, I told you that we had a genetic blueprint. Think about it in a sense like this. So let's say you have a building and you have flats that are one above each other, and then they're organized in a particular map-like structure such that the kitchen is always reliably above every kitchen in the building. You have a blueprint that the architect has used to build this building. And yet in the detail, one kitchen differs from the other, and that, the masala dabba will be in a different place. The fridge is perhaps placed slightly differently. There are a whole bunch of details that will be different, so that even if you went in on every floor of the building only to the kitchen would not look exactly the same, even though the walls, perhaps the sink, are in similar locations. Much the same way, even though you and I have an occipital lobe, which is in the same place, our primary V1 is in the same place, in the detail, we will vary. Okay. For example, let's say one of you in here is a concert pianist, or someone plays the violin. Well, their somatosensory cortex, in particular for their fingers, will have a very different representation from that in my brain. I don't play any musical instrument. And that's just to tell you that every experience that you have instructs this blueprint. And so this blueprint is not a hardwired structure that is just going to say exactly the same forever. It changes and it is tweaked by life experiences. And in a sense, this is what I refer to when I say instructing the blueprint. So this term that I want to use, and I'll come back to multiple times, the term called neuroplasticity is a sense that you have the ability to change what is there. And while you may not change landmark things, you're not going to be replacing your occipital. One person's occipital lobe will not move from one location to the other. In the detail, you can start seeing a fair bit of inter-individual variation. And this is also different for you yourself. So it's not just inter-individual. Even you as a person going through across your entire lifespan are going to see changes in detail. And this is how the brain constantly talks to the environment and incorporates information from the environment to undergo change. And experience is really the greatest educator for the nervous system in that sense. So I've told you that experience will shape your brain. Now, let me ask you another question. Do you think it matters when? Right? I mean, is experience an equally powerful educator throughout your entire lifespan, or are there windows in which you are most prone to taking information from your environment? Well, the news is that it is downhill in that you tend to take the most amount of experience evoke change to your brain in the earlier windows. Okay. Not to say that when you're old and you're in the aged pool or you're middle age like me, that you're still not incorporating change. Of course you are. And that's our ability to learn and change and acquire new skills is something that's lifelong, and we can keep putting it into use. And yet there is no question that the scale and the grandeur to which plasticity can happen in your brain does shrink with time, and it reduces as you get older. It's most dramatic in these windows of life that we call critical periods. So I'd like to define this term. I've defined plasticity for you. Now I'm going to define critical period plasticity. I still haven't told you how plastic changes happen in the brain. I haven't told you what do I mean by plastic changes. I literally don't mean plastic in your brain. I mean the ability to change, right? So the ability to add or remove synapses, the ability to change the strength of a connection, either to increase the strength or decrease the strength. What do I mean by strength of a connection? When a neuron fires, how reliably or how strongly does it cause the corresponding postsynaptic partner to fire or not fire? That is the strength of the link between two neurons in a sense. And that strength can change. And that's how synapses actually undergo what they call a gain in strength or a reduction in strength. And, one neuron can have something like 10,000 synapses. So this will just give you a sense of how this can then scale, right? Okay, so let's come back to the idea of critical period plasticity. So these are time windows during development when the brain is most sensitive to experience. And perhaps I'd like to draw on ideas from Darlene Francis and others who have put forth something called a tuning hypothesis. Think about it like your brain using this period of time to predict what it's likely to see. Okay. So you built the framework using a genetic blueprint. Now you want to tweak this blueprint. So what are you going to use to tweak and determine the most optimal blueprint that's going to allow this brain to function most optimally for the environment it is likely to see? You use the early windows to sense the kind of environment you will have to prepare this nervous system for. And so there is this tuning hypothesis, very nice idea that suggests that you actually adjust the function of this framework and blueprint that has laid down these neurons and connections to predict what you will likely have to deal with or handle. And based on that, you retain or strengthen or reorganize the nervous system, right? So these are critical periods. And it's really the critical period that's allowing these circuits. And what do I mean with a neurocircuit? The connections between populations of neurons. So for example, the visual nervous system that I was referring to is really the eye connecting via the lateral geniculate nucleus in the thalamus to primary V1 in the occipital lobe, and then multiple other connections that are made. So this whole pathway is the neurocircuit of vision. And it's a very minimal circuit that I'm articulating, but there are, of course, many broader components and lots of crosstalk that is beginning to happen. And based on the nature of visual input you get, you will modify this environment. So for example, let's imagine very classical experiments done by Hubel and Wiesel, where if you close input from one eye, so now you don't have input from one eye during these critical periods, there's really nothing wrong with your retina. Everything's fine. You can actually see light. It's just that you've not allowed light in by keeping this eye closed. And there are ways in which you can do this experimentally, where closing, suturing, blocking, occluding this eye prevents any input from coming from that eye. And that eye projects to the opposite occipital lobe. What happens is you lose, via binocular organisms, you lose territory. You actually lose the part of the brain that is responding to this eye, because in a sense, the brain is saying, that's actually not a very valuable input. Nothing is coming from there. Let me dedicate more territory to the other eye, right? And if this happens during very critical early developmental windows, you will end up becoming functionally blind in that eye, though there's really nothing wrong with the retina, right? And if you open the eye after the critical period, you cannot regain or restore fully functional vision. But if you open it during the critical period and allow experience to come in, it can rewire. So this is the critical part that I want to tell you about, that there is change and there's dramatic change. If you and I occlude our eyes in adulthood, nothing happens. This occlusion works only during these critical windows. So there are critical periods for all kinds of modalities and all kinds of function, and they don't all overlap. They have different windows. They're different. The strength of the critical period can have it can have a broadish framework, and there's a very hard and fast critical period with sort of fuzzy endings in a sense. Let me use language as a good example of this. Okay, most of us who are listening to the seminar, most of us who are in India, are multilingual, bilingual for sure. Most of us are multilingual. We speak multiple languages and we flip back and forth between languages with great ease. Partly not because we were taught these languages in any pedagogical manner in school, but because we listened to them growing up and they were just multiple languages spoken around us. So language as a skill is something that's actually, as a critical period window for language, is pretty early, right? And it starts closing in somewhere in the adolescent window. Now think about when we bring in multiple new languages in teaching processes and in childhood training and learning. Very often, we're not teaching multiple languages at the early stages, and we are focusing on one language, worried about whether full language development will not happen if there are too many languages as a confusion. And that's not necessarily the case at all. In fact, if you now come in with foreign languages or new languages later, it becomes harder and harder to learn them. All of us know this. If today you and I decide that we want to learn a new language, we're going to really struggle. The critical period is closed. It's not to say we won't learn it. It will just be a ton more effort and a ton more work to acquire the same degree of proficiency that a young kid who's just 7 will just 5-6-year-old will just pick it up with great ease if they're constantly bathed or exposed to the language. That's why I'm using the idea of language. But really, there is much in here, and the emotional control window is also going to be critical in this regard. All right, so I now want to move from work that, of course, has been done in sort of clinical and epidemiological studies in humans, but to move to what we can study in animal models that might inform us and that would allow us to go back and forth between what is seen in the clinical epidemiological context and what we can do in a more sort of detailed molecular cellular way in animal models, which is not possible, of course, in the human condition. So we do know, and we know this well from a diversity of preclinical studies, epidemiological studies, and clinical studies, that early environment has a profound effect to play in shaping brain function and adult behavior. We also know this for emotional behaviors. We know this in terms of also risk for psychiatric disorders. So several studies indicate that early adversity, early trauma is a common risk factor. I gave you the example of the visual nervous system because it was easy for me to pinpoint a part of the brain one could perhaps focus on, this blue part, the occipital primary V1 area, because we know where it's located, we know what it does, we have a far better understanding. It's still incomplete, but a far better understanding. Where do you start looking in the brain when you want to look at things that regulate emotion, circuits in the brain that regulate emotion? And how do you define emotion in the first place? What set of behaviors fall into the category which is broadly and loosely defined as emotion? Well, certainly social behavior, anxiety, fear, despair, attachment. I mean, it's a very wide spectrum of behaviors that will fall into this category. Where would you look? Is there a defined single circuit in the brain that says this is the hallmark center for emotion? No, there isn't. Unlike for vision, where you will start at least by saying primary V1, LGN, then from there to V2, V4, V5, other areas, association cortices, it's much harder to pinpoint a clarity of hierarchy of structures in the brain that regulate emotion. We have a reasonable sense of who's involved. We do not yet have a sense of the hierarchy and all the players. And we have it for a limited spectrum of emotions, partly because this is limited by what we can study in rodent models. fMRI and imaging studies in humans provide insights, and certainly studies in primate models, non-human primates, also provide insights. But they are limited in the detail to which we can give you a granularity of which population of neurons. So, for example, if you say, is laughter an emotional state? Is the ability to feel, is humor something that is associated with an emotional state? Very hard to give you a sense of which circuits are directly implicated, even though we have a broad sense of the parts of the brain that light up when one is, appreciating humor, for example. But humor, of course, also has a big sociological and cultural context. And so I want to tell you that this is a layer of stuff that will emerge in humans, which is far less removed in the rodent models that we study. And this granularity that you need to overlay over information, sociocultural contexts, which are so critical when we study the nervous system, sometimes disappears a little bit when we study it in rodents. And this is something as a caveat to keep in mind. All right, so I'm caveating by telling you there are multiple circuits. The hippocampus is involved, the prefrontal cortex, the accumbens, the amygdala, the ventral tegmental area, the hypothalamus, the raphe, the LC, and I'm just touching the tip of the iceberg. There are many, many circuits in the brain that contribute to the regulation of emotional behavior. Let's come to why it's important to study this in the first place, right? Here is the work from a very large study that was done in a managed healthcare in the US, out of Kaiser Permanente, which is a big managed healthcare group in the US. Study done by Felitti et al. With a population of about 17,000 individuals. So a very large study, which was questionnaire based. And then you were using that as a way to follow people and see what was lifetime risk for depression, substance abuse, anxiety, obsessive compulsive disorder, other health-related behaviors, etc. What emerged was, if you quantified the number of adverse childhood experiences, and this is questionnaire based, and if you see that the number of, so 4 plus is 4 reported examples, it could be social, it could be sexual abuse, physical abuse, it could be neglect, a variety of measures that were being measured out. When you see this and you're here scoring for a lifetime history of depression, you can clearly see that there is a higher risk in individuals that have had more than one adverse early childhood experience, event of trauma in early life. And there's, of course, also a sexual difference in this particular measure, and that will vary. For example, if I was to show you substance abuse, once again, there's a greater risk in individuals who've had early adversity, but the sex ratios flip. So the predisposition is higher in men than it is in women. And then to put this into a broader sociocultural perspective, this will matter where and which population you're looking at. Because, for example, in the Amish, where world over you have depression rates which are higher in women than in men, in the Amish where substance abuse or, for example, access to substances of abuse is limited, the depression rates actually don't look very different between men and women. And there's interesting studies that have come out that suggest that you must keep these factors in mind when you're looking at even this nature of epidemiological data, because that highly depends on the environment as well. So there's an increased risk of substance abuse, anxiety, hallucinations, depression, OCD, spectrum of disorders. And this is a slide that I've taken from Martha Strauss's presentation, where I just want to define trauma, early stress, etc., right? Because it's a continuum. So everyone goes through stressful experiences in life. The question is how mild or how easily controllable or how easily can you surmount those stresses. And that, of course, also varies at an individual level. So there is no hard and fast boundary of positive stress, tolerable stress, and toxic stress here. In fact, it's a fluid continuum which will vary based on the individual that is involved. And yet it's clear that there are stresses that we all cope with routinely and that we handle them really well. And in fact, they are part of the training of our environment, working on our stress response pathways to tune them appropriately. Okay, so there's that element of what we call eustress or stress we cope well with. There's tolerable stress, which is stronger And yet, given the right coping mechanism and the right support structures, you will see people surmount these kinds of stresses as well. And then there is sustained stress for which it is difficult to surmount, difficult to override, and carries long-term health costs, right? So this is something that is interesting to think about. And really, these boundaries are fluid, vary greatly from individual to individual, and there is a context that plays a very, very important role. Okay. And as ACE scores increase, which is early childhood adversity scores, you can see that the risks go up, and the number of people who are, will likely exhibit health-related risks is expand significantly, going up on the right hand side. Just want to remind you that when we often think about early adverse experiences, we tend to think about physical abuse, sexual abuse. Poverty, by the way, is a big factor as well. But neglect is one of the most common forms of child maltreatment. This is data coming out of the US. Be very relevant to look at what in India constitutes early adversity, because poverty will come in as a major player, and it is absolutely a major player. All right, so now we've discussed why it's important to study this. There's no question it's a key player. It's a key player from the point of view of public health, key player from the point of view for scientists, also for all taxpayers, from what you see going forward for your country. And for India, with a very large young population, this becomes extremely critical because it will shape many, many things for the country in the decades to come, and in fact, in the century to come. So how do you model this in animal models? How do you even study this, right? I mean, we have used terms like abuse, neglect, trauma in the context of the human population. How do you start studying this in rats and mice? The species I use, which are laboratory rats and laboratory mice, are not biparental naturally. And so they tend to have only one major caregiver, which is the dam. This is a fact you have to keep in mind. And so a lot of the models that I will use will perturb interaction between the dam and the pup. There are other species in which it is both mother and father, and there you need to perturb interactions with both. And then there are species in which it is that it's not as small as that. Non-human primates, for example, it's a community. And then it requires disruption of its neglect at the level of the community. So it's not just one individual at all. So keep this in mind, that when I'm talking about models, when you are extrapolating, you cannot do a straight out extrapolation, because these species are very, very different. And yet the nervous system, you can understand many things at the level of the nervous system using these species. Just be careful when you go straight in a extrapolation. All right. So in these models, because it's really only one primary caregiver, which is the dam, the mother, you can look at inherent differences in maternal care. And this is work that's come from Michael Meany's lab at McGill for several decades now, doing beautiful work on looking at inherent natural differences in the quality of care. And then there are more perturbative models where you can go in and actually perturb the behavior that the dam shows towards her pups by taking away the pups for short durations, which sort of fragments the kind of maternal care that they get. And then that changes the kind of care she administers to the pup in a laboratory setting. Okay, not in a natural setting, but in a laboratory setting. Be aware of that. Okay. So many people, and I'm summarizing work from many groups all over the world, have used many ways of going in and modulating experience during these critical periods, which are during prenatal and postnatal life. Okay, I will predominantly talk today about postnatal experiences, not prenatal, but there's a huge component of the immune milieu, the nutritional milieu that happens prenatally that can also shape the way the brain develops and how these circuits respond in adulthood. Okay. And the way in which people have done it is by looking at models in which you can look at natural variations. You can take pups away from their dam. You can also enrich the environment, change the complexity of the environment, and look for positive interventions that may have beneficial effects. You can go in and perturb specific neurotransmitters or specific circuits in the brain, and I would share that data with you. And when you start looking at all of these models, what emerges is that common across models of diverse models of trauma or adversity is that when you look at these animals and they become adults, in the kind of behavioral ethological tests that you do with these animals in the animal house or in the laboratory, where we measure anxiety-like and despair-like behavior, I'll show you some of those tests so you get a sense of the kind of tests we are using to study these behaviors. You can look at hormonal stress responses. You can look at the ability of the animal to learn. You can look at social interaction between conspecifics of this species. You can look at architecture in the brain. You can look at the ability to produce new neurons. You can look at the natural process of aging. You can look at how they buffer stress. All of these responses get altered when there has been early trauma. And many of these get altered for the rest of the life of the animal. It's not hard and fast. You can come in and intervene and change it. And yet what emerges is that trauma in this window has very long lasting consequences, whereas the same kinds of stressors or stressors in adulthood are often short-lived, very quickly surmounted, and the animal comes back to baseline very quickly. So stress matters as far as these measures are concerned. It matters the timing of the stress is equally critical. So here's a model that I described, which is one which was set up by Seymour Levine, Paul Plotsky, and many others. This model was set up actually in the 1950s. And what it involves is taking the pups away from their dam for 3 hours a day, which is not a very long perturbation because the dam might actually forage in the wild for food for that kind of duration. So it's not unheard of for her to leave her pups for this duration. However, in a laboratory setting, she's not choosing to leave. You're taking the litter away. And when you return the litter to her 3 hours later, so they're taken away, they put on a euthermic pad, they are maintained, so the thermoregulation is maintained, they come back to the dam. What happens is over a period of time, when you do this every day for 3 hours from postnatal day 2, so 2 days after birth till 12 days, pups are developing normally, no nutritional deprivation, and yet the quality of nurture. So you can measure the number of nursing bouts, the number of sort of, arch back, licking, grooming, etc. seems to deteriorate. And that's been one of the hypotheses suggested for why this model evokes very long-lasting changes in anxiety, stress responses, metabolism, cognition, not just in the brain, but also in the periphery. It's a model for irritable bowel syndrome because it has effects on the gut and the periphery as well, changes the production of newborn neurons in the brain, changes the serotonergic pathway, which is what I will tell you about today, leaves many epigenetic consequences such that multiple genes within their promoters show changes with chemical tags on the histones or directly at the level of the DNA, which will change the expression of those genes. So this experience is in a sense marking the manner in which certain genes will express proteins. Okay, so going forward, you're carrying the marks of this fairly long lasting, at least out till 20 months, long after the experience is over. You can mimic a lot of this by actually giving a pharmacological drug that elevates levels of serotonin in the brain. And now this is paradoxical because this drug, fluoxetine, is actually the drug Prozac, which works as an antidepressant and works to reduce anxiety. That's how it works in humans. That's how it works in non-human primates. That's how it works in rodent models in the adult. However, when the baby brain, either postnatally or intrauterine, is exposed to elevated levels of serotonin during this developmental window, it has exactly the opposite effect. It ends up increasing anxiety, increasing despair-like behavior, perturbing cognition, changing a bunch of things for the lifespan of the animal. So just tells you that just because it's a drug that is used as an antidepressant, it's about the timing and it's about when the individual is exposed. So it does matter. This is really relevant because it's a drug of choice for women who have gestational depression, women who have postpartum depression. It does cross through from breast milk. It crosses through across the placenta. And it raises questions about exposure in the child. And, epidemiological studies are clearly required to extensively look at this. There's already clinical data that indicates that exposure to this drug in children and young adolescents increases the risk rate of suicidal ideation. And that's something to keep in mind, that a drug, just because it works a particular way in the adult brain, does not necessarily work the same way in the developing or the modulating plastic window of early postnatal development. Here's work that's actually also from a slide taken from Martha Strauss, work from the Radley and Bock et al., which shows that persistent stress in these developmental windows can bring about architectural changes in the brain as well, where the synapses, and you can actually see spines in prefrontal cortical neurons, which are actually deteriorating and disappearing. And so you can see damaged architectural shape and spine density in these neurons in both the prefrontal cortex and the hippocampus. Why will this matter? These circuits, the prefrontal cortex and the hippocampus, are top-down controlling circuits. They are circuits that provide context and allow you to switch off stress and control and modulate the degree and the extent to which a stress response will be mounted. They matter not only to switch off the hormonal stress response, which is so essential for your survival, right? It switches off digestion, reorganizes your immune response, really switches off, for example, sexual activity and behavior, so dampens the libido. It drives more glucose to your muscle, which is so critical if you had to mount a sort of a flight response. All of those are critical and they are important for an effective stress response. But if you keep having them on or you don't switch them off appropriately, then you're going to have a bunch of other things that emerge which are not so great. Okay, these are what happens when the stress response is not feedback modulated. There are executive functioning areas of your brain that turn off the stress response. So as important as it is to switch it on, it's equally, if not more important, to switch it off. And it's these circuits that actually drive shut off. Well, these are the regions in which you see damage when there's been sustained early trauma and early stress, and the ability to modulate the stress response is what goes awry in the brain, in a sense. So I will be telling you today about work we've done in the prefrontal cortex of rodents. And we're interested in the prefrontal cortex in particular because this is the circuit that talks to multiple top-down circuits, talks to the amygdala, which drives fear responses, the hypothalamus, which drives the hormonal stress response, the raphe in the midbrain, which drives perception of control over stress. Very, very important to have a sense of control. It is that perception of control that immunizes you, in a sense, to be able to handle the stress. And when you have a feeling of an uncontrollability over stress or an inability to mount a shutdown of the stress response, that can have its own problems. And of course, circuits that are associated with reward and pleasure seeking, which are also modulated during stress and can really impact recovery and sort of hedonia related behaviors. All right. So Madhu was a graduate student in the lab at that time. I'll tell you about one set of stories which emerged from my lab over the last decade or so. And so I'll share that work with you. What Madhu was a graduate student wanted to ask was, is there a change in the function of the serotonin 2 receptor in maternally separated animals? We'll just focus on this one story. I mean, we look at multiple things, but this is one example I want to share with you. The serotonin 2 receptor is abundantly expressed in the cortex in the brain. It's a receptor that is targeted by a variety of drugs that are used to treat schizophrenia, that are used to treat depression. It is also a receptor targeted by hallucinogenic compounds, psychedelic drugs like LSD, ayahuasca, psilocybin, which is a key component of magic mushrooms. So it's also a receptor that drives hallucinatory responses. Receptor of interest to people in the psychopathology, psychopharmacology field. So Madhu was not the first person to think of this idea by no stretch of imagination. There were many people who had obviously addressed this. Perhaps her take on it was a bit unique in that what she decided to do was not ask, is the level of the receptor expression changed? Or do you have more receptor or less receptor? Or is it different in different parts of the brain? What she wanted to ask instead is the function of the receptor changed? And I'm glad she started that way, because if she had simply looked at levels, we would have stopped. It would have been a single experiment. Nothing has changed with levels of the receptor. Yeah. So what Madhu did was she said, I'm going to actually, along with students like Sthita, Ankit, and Amrita, decided to ask, can I look at a behavioral response that is driven when you stimulate this receptor? So we have pups, we separate them every day from P2 to P14 for 3 hours, and then they go back to the dam. And then they're back with the dam from P14 onwards and left undisturbed until they're weaned about P21 or P28. And then they live along with their same gender mates, litter mates in an animal house cage, undisturbed until we do these experiments, other than for animal facilitated rearing, where of course they're provided food and air conditioning, water, all of that. So what you see is that animals, when we test them for behaviors at postnatal day 21, so when they are 3 weeks old, which is literally a week after the trauma is over, and then 3 months later, so this is now when they are actually 3-month adults, they're full-fledged adults, and we're looking at behavior in response to stimulating this receptor. So I told you that this receptor is also stimulated by hallucinogenic compounds. So we use a hallucinogenic compound called DOI, and this evokes a very stereotypical head twitch response, which is classical and seen in rodent models in response to all hallucinogenic compounds like LSD, etc. Very easily measurable response. The animal shakes its head in a very stereotype manner, like it's actually everybody has seen this response in a sense. You would have seen it if a dog gets its head wet, right? Any dog that has its head wet has a very classical way of shaking off the water. It's called a wet dog shake, or there's a head twitch, sort of a head shaking behavior. That is driven by the 5-HT2A receptor, and hallucinogens increase this particular behavior quite drastically. Very easily measurable. In fact, we are blinded to the treatment groups, and then we just record. Multiple people record the behavior, then you analyze it, and what you see is here are control animals. They had no disturbance in their early life. When you give them the drug, whether they are 3 weeks old or they are 3 months old, you get this very clear increase in head twitch behavior, significant induction, and this behavior is very stereotyped and you can measure it. What I want you to note is what happens to animals that went through this early stress of maternal separation. For the same dose of the drug, when they are 3 weeks old or 3 months old, you get a stronger response. And so what Madhu was saying is that if you had a history of trauma, for this particular receptor, the 5-HT2 receptor, you're actually ending up with a stronger response, right? For the same dose of the drug. So you don't have more receptor, but you just end up with a greater response to the same dose of the drug. That was really interesting to us. So then we wanted to show this in multiple ways. And in collaboration with Evelyn Lamb, Madhu went to her lab and to University of Toronto. You can take slices through the brain and you can actually record directly the amount of current that is being generated in these neurons in response to the drug. What we're measuring is 3 micromolar exposure of these brain slices to the drug, the degree of response in control animals, and the degree of response in maternally separated animals to the same dose of the drug. What I've shown you is behaviorally you will get a stronger response. Much the same way, the inward current is significantly greater in animals that had a history of stress. Why is this happening? You don't have more receptors. So why are you getting a stronger response? Turns out this receptor is coupled to a strong signal transduction pathway of multiple enzymes, and you can measure the activity, the expression of these enzymes, etc. And just to cut a long story short, we looked at it first agnostically to see which genes are changed by looking at all genes expressed in the prefrontal cortex and the hippocampus. And we find that the whole pathway that is downstream of this receptor ends up potentiated, right from the coupling to the G protein GQ to many, many enzymes that are downstream. So the receptor itself is not changed, but many things that are going to be downstream of that receptor are just higher in the brain that has undergone maternal separation. And you can see this in terms of the nature of activity produced in the brain. And here you can measure it by looking at the gene expression of an activity responsive gene. So this gene increases the expression of the mRNA for this gene every time that a neuron fires. So when neurons are firing particularly heavily, you will see greater expression of the gene. Here are controls. So I'll just walk you. It's a busy slide, but just look at the control. So control has nothing on board and there's really no real expression in the brain. Maternally separated animals actually baseline don't differ from control. Here are control animals given the drug DOI that stimulates this receptor, and obviously stimulating the receptor activates neurons. And you can see this very cleanly by an increase in gene expression. But here are maternally separated animals given the same drug. And what you can see is that for the same dose of the drug, the nervous system responds very differently when these animals have gone through trauma. Right? And that you can quantitate, and we've looked at multiple such measures. So what I've shown you is that for this receptor, and here's an example of one, but we've looked at many other receptors as well, and some other receptors, Gq-coupled receptors, are also changed, and this becomes relevant a little later in the talk. What I've shown you is that going through trauma will change the way you recept this receptor for the serotonin, for serotonin, so this is a serotonin 2A receptor, ends up with just altered response, right? Don't have more of the receptor, don't have less of the receptor. Receptor levels, if you measure, are just identical. And yet the response for the receptor is potentiated and enhanced. And this becomes particularly interesting. I won't go into why we think it's happening. So Madhu said, okay, this could just be one of n number of receptors. So serotonin has 14 receptors. So how do I know this even means anything, right? Does this actually matter to the kind of behaviors this animal is going to show in adulthood? So she decided to block this receptor using a drug, ketanserin, which blocks this receptor. And what she did is she fed it to the animals right before they were separated. So she would take an animal out, give them a drug that blocked the receptor, then they would go through separation, then they would go back to the dam. And she block this receptor in the entire window during which the separation and the trauma was being done, experiment. So we would have 4 groups. The groups are the following, and this is a busy slide, but I'll walk you through the data so you get a sense of how we do these experiments. There are 4 groups. Each group would have an N of about 15 animals per group. So they're talking about an experiment of about 60 animals. You have a control group, which doesn't go through maternal separation and is fed a vehicle. Vehicle is the sort of solution in which we dissolve the drug, right? Then you have a control group, doesn't go through maternal separation, fed a blocker of the 5-HT2 receptor. You have a group which is maternally separated, goes through maternal separation, but is fed just the vehicle, so no drug. And then you have the combination where you have the maternal separation, but you block the 5-HT2 receptor. Once you do this experiment, then this happens from postnatal day 2 to 14, then these animals become adults, and then we run them on a battery of behavioral tasks for anxiety, despair, cognition, etc. Like spectrum of things, and we're also looking at changes that are happening in the brain. So we have lots of such cohorts that are going through it. And when you look at their behavior, I'm going to just give you an example of this task and see. So you'll see these squiggly lines and wonder what they are. They're actually a representative from one such control animal that was put in an open field, which is a chamber, where we have an infrared camera watching their behavior over a period of 30 minutes or sometimes over a period of 10 to 15 minutes. And you can see that the animals like to cling to the corners, and that's not surprising to any of you who has had a rat or a mouse in their house. You would know that the rat is not going to come into the middle of your living room and sit in the center ever. It'll run along the corners with this thigmotactic behavior where they like to stick to the wall and then run along the sides, right? And then quickly escape into a hole. So, well, animals in an animal house, when put through this task, will also prefer to spend times on the periphery rather than hang out in the center. So you can actually measure the distance they cover, the speed that they cover, how much time it is, because we are recording it for every animal. We can actually trace down every bit of behavior that they show. And you can see that the animals enter the center less, they spend less time in the center, etc. So you can actually measure this. And animals that go through maternal separation show anxiety or fear-like responses. They enter the center less, they spend less time in the center. This is just 2 measures, but we do it on a battery of variety of measures. And then you ask, okay, what happens to those animals that have gone through this trauma, but we didn't allow serotonin to work on the 5-HT2 receptor? When you do that, and you block the 5-HT2 receptor, you rescue the behavior. Remember, the behavior is being studied 3 months later. All these trauma events, drug, etc., has happened in the early developmental window. Then you leave them alone. You study the behavior 3 months later. And this is something that she saw multiple times. I just want to give you one more example. And this is now looking within the brain. So now what have we done? Again, there are 4 groups. You have a control group, which was fed vehicle, went out into, actually there are 8 groups, sorry, not 4 groups, 8 groups. Control group fed vehicle, they go into adulthood and they're left undisturbed, and we look at the brain. Then there's a control group that was fed vehicle, goes into adulthood and now sees chronic stress, okay, in adulthood. So the early trauma and a second hit of stress in adulthood. And then you have a control group that was fed the blocker, went into adulthood, either saw a normal environment or saw stress. A group that was maternally separated, so early trauma, fed vehicle, no drug on board, went into adulthood, saw stress or no stress. And then a group that was maternally separated, fed the blocker, went into adulthood, stress or no stress. So 8 groups, right? Let's walk through the top panel. What you will see is now activity-dependent gene expression for ARC, which will tell you about the activity in the brain in adulthood in response to the stress based on your early life history, okay? So control animals don't really have anything going on. I mean, there's some amount of baseline expression in the prefrontal cortex. Maternally separated animals look just like controls. Animals that have gone through stress for 10 days have totally adapted to the stress. And this is a restrained stress in which the animal is kept in an environment where it doesn't have free movement ability. So it is a little restrained. It's like going in the Mumbai local. That would be our equivalent, but we adapt to it, right? We do this routinely and none of us worries about it. So much the same way the animals adapt beautifully. First time they see it, they're stressed. After that, they just adapt. And you can see the adaptation. There's no activity in the circuit at all, unless they went through trauma. If they went through trauma and then they see the stress in adulthood, they respond to 10 days of stress like they're seeing it for the very first time. And that's most dramatically visible here. You can see it in this panel. You can see it in this panel. It's really pretty drastic example of this. You can actually see how much these animals fail to regulate their stress responses. except when you block the 5-HT2 receptor, it comes right back to normal. And that was really interesting to Madhu and us. So we knew now that this receptor is important. Its function is altered. It's important for the development of normal central stress responses. It's critical for the development of anxiety-like behaviors in adulthood. I'm just going to skip through some of this part because in the interest of time. And what we found is that this is not a receptor that's not only important in the maternal separation model, it's equally important in a model in which you elevate levels of serotonin. When you allow serotonin to go up, but you block this receptor, you do not produce anxiety and despair-like behavior. And that's particularly interesting because we now know from this work that if you simply go in and stimulate this receptor, just selectively stimulate this receptor, you will produce enhanced anxiety and despair-like behavior in adulthood in these models. Why is this clinically interesting? Because the function of this receptor is known to be altered in the platelets and the periphery of patients that have psychiatric disorders with a history of early trauma. And that's why this receptor becomes so interesting and clinically relevant. So what Sthita then decided to do in the last few years, and I'll quickly wrap up this story, was to ask, is there a theme here? Is it that in response to early trauma, the brain actually drives up GQ signaling? GQ is the G protein to which these receptors are coupled. I gave you the example of the 5-HT2A receptor. We also know the muscarinic M1 receptor is perturbed in these animals. So is it multiple such receptors that are driving more GQ signaling in the brain? And because you drive enhanced GQ signaling in these very critical windows in specific parts of the brain, you end up producing altered behavioral states, altered sensory motor gating, which is a highlight of what's seen in schizophrenia models, altered despair-like behavior, increased anxiety, increased vulnerability to stress. So that was the question he set out to ask. Now, how do you go in and selectively perturb GQ signaling in a select part of the brain in a subset of neurons? You could not do this easily in the past. Technology now allows us to do it. We can genetically modify and introduce genetically modified receptors only in a subset of neurons, here in the excitatory neurons of the forebrain, using a chemogenetic tool, which will allow us to enhance GQ signaling in response to administering a drug, which selectively stimulates these genetically modified receptors. So essentially, it's a way for you to go in and perturb GQ signaling in the circuit. And when you do that, in this window, in the cortical neurons and forebrain excitatory neurons from P2 to P14, which is what Sthita did, and then go in and look at behavior in adulthood. And this is just proof that we have stimulated that GQ signaling pathway. There are multiple ways for us to measure it. This is just showing you that you actually get the genetically engineered receptor into the right population of neurons. It causes those neurons to fire. We can activate GQ signaling in these neurons. And when you do that, normal development is not perturbed. They put on weight regularly. They show normal reflex behavior development, which tells us that those things are happening absolutely normally. But when you start looking at their behavior, then in adulthood, they show anxiety-like behavior. They show despair-like behavior on a variety of different measures. And they show, this is shown in multiple ways. Here's another, using another drug to drive GQ signaling in this developmental window, capitalizing on these mutated receptors we can express. Here's on the elevated plus maze, which is another behavioral task. And you can clearly see strong anxiety-like behavior. They also show perturbed sensory motor gating, which is a hallmark signature of schizophrenia-associated behavior. Pre-pulse inhibition in these animals is utterly perturbed in 3-month-olds who had this kind of a stimulation of GQ signaling in the early developmental window. It's all timing, because if we do this when they are juveniles and adolescents, or when we do this when they are adults, it doesn't have any such effects, right? So timing is critical. Matters when you go in and perturb. That window is the critical window. We don't know exactly when that critical window ends. We definitely know it's earlier than P21, but is there a window later than that? From P28, which is postnatal age 28, so 4 weeks onwards, we don't see any such effects when we go in and perturb. All behaviors are normal, right? So this is interesting. This allows us to start figuring out which population of neurons. So right now, if you look at the complexity of the circuitry, we have perturbed excitation in the principal forebrain excitatory neurons. When we perturb GQ signaling and excitation in this population, we produce anxiety and despair-like behavior and schizophrenia-like behavior way out, months and months later. What happens if you perturb it in other populations of neurons? And these are kinds of experiments that we can now start doing. Can we reverse the effects of maternal separation if you go in and inhibit specific circuits? How do we actually titrate this back and forth so we start understanding the circuitry that is driving these behavioral changes? I'm going to skip past this in the interest of time. What I've shown you is work that led from first identifying that one GQ-coupled receptor, the 2A receptor, was perturbed. It was important for establishing anxiety and despair-like behavior. From there, we went to the next level to ask whether the GQ component, which is GQ signaling downstream of this receptor, drives these kinds of behavioral changes, and use these tools in mutant mouse models now to start studying this. And now next natural step is to ask, can we reverse these effects? If we have to reverse this, do we have to do this while the animal is young, or can we reverse it later? How easily reversible are these changes? Are they hardwired so that it's very hard to reverse them? How easily can we reverse them? Do we need to reverse it with drugs? Can we reverse it with environment? Can we reverse it with exercise? What are the interventional strategies to actually reverse some of these changes? And so I've shown you data that maternal separation and postnatal fluoxetine perturb this GQ-coupled receptor, the 2A receptor, that drives anxiety and despair-like behavior. Simply stimulating this receptor is sufficient to mimic many of the effects of early stress and trauma and reproduce anxiety and despair-like behavior in adulthood. We know that forebrain excitatory neurons and GQ signaling in these neurons in this early developmental window is critical. In this circuit, when you have aberrant or enhanced GQ signaling, you actually program enhanced anxiety, despair-like behavior, and certain hallmark signatures associated with schizophrenia. We'd love to ask if you can do the reverse, which is, can you promote resilience, reduce vulnerability, and how easily can you reverse some of these changes as well in these animal models? We study this at a molecular level, cellular level, synaptic, circuit, and network level, with our idea that we want to study these circuits to understand how behavior is changed and to be able to draw connections between these levels. That's easier said than done. We would love to be able to draw the links across all these levels. And that's something that the community struggles with as a whole, but we try. Just skip this. So the focus of my lab is to ask two central questions. One is to ask, how does life experience directly modulate neural circuits to change behavior? How does it do it at the level of specific receptors, specific neurotransmitters, specific pathways? Can we understand this so that we could actually design ways to tweak it? And tweaking it may not only involve drugs. Tweaking could also be environmental strategies that can reverse these effects. We're equally interested in the circuits and molecular pathways that are downstream of pharmacological antidepressants, but also environmental strategies that can reverse some of these effects, like exercise, enriched environment, caloric restriction. And this is the amazing team that I'm very honored to work with, whose work I was able to share some parts of the work with you. This work is done at TIFR. Some of the work that I described today involved key collaborations with James Chelaya at JNCASR and Anant Patel at CCMB in Hyderabad. These are my acknowledgements. I would not be able to do this work without a very talented team of graduate students and postdoctoral fellows whose work I described, some of the work I described. Sheetal Suryavanshi, who's the person who heads our Animal House facility and manages all these diverse lines of animals. Collaborators, funding that comes from TIFR, DAE, and the PVF grant, which funds our research. And I'm more than happy to take questions. Sorry if I went slightly over time today. Happy to take questions.
All right. Thank you so much, Vidita. That was very insightful. We have a comment on Twitter. Kirti says that it was very insightful. And then we have a question here in the chat box. I'll just read it. The blockers you use to block this receptor, is there any evidence of how specific it is to this particular receptor, or can it have off-target effects? So that's a question from Supriya.
Good question, Supriya. Everything always has off-target effects. By definition, there's not a single drug that won't hit something else or the other. The question is only just at what dose and when does it get into the zone where you're at a high enough dose that you're going to start seeing a bunch of other things that it is hitting. Ketanserin hits the 5-HT2A and 2C receptor, but it also blocks histamine receptors. At the dose range at which we are, we believe most of it will be through the 5-HT2A receptor. There are other ways we can show this by actually looking at, so we've done it with a bunch of drugs. I gave you data with Ketanserin. We've used an even cleaner drug, MDL-100907, which blocks this receptor even more selectively. But even with both of those drugs, the thing with all of this is that when we block that receptor, we also block the effects on those neurons which secrete other things, other neurotransmitters, which have their own downstream effects. So we may be blocking one end, we cannot preclude the possibility of effects that are also happening on other receptors as a consequence of that original blockade. So while we believe it's the 2A that's the most critical, I would certainly not rule out effects on other receptors as well. In fact, we already know that the muscarinic M1 receptor is itself perturbed. Other Gqcoupled receptors are likely to be perturbed. Calcium signaling in the circuit is perturbed. So it's likely to be broader than just this receptor. Yeah.
Okay, I'd just like to reiterate that we have started the Q&A session, so please feel free to put in your questions in the chat box. Debirati has raised hand. Debirati, over to you.
Yeah, thank you so much, Professor Vaidya. It was an amazing talk and sort of a refresher for me because I was a postdoc in Shona's lab at NCBS for a brief time. So, thinking about stress and receptors and blockade, brought back fond memories. So right now, I am, public health researcher, and we are asking a very similar question in the sense that we are we're trying to identify how or what basically are the early life factors that affect neurodevelopment and children's mental health in the first 10 years of their life. So, we recently got funded by the India Alliance to start the project this year. So we'll be looking at many different factors, nutrition, psychosocial and environmental pollutants, and looking at, the interaction of all these factors and how they affect children's brain development and mental health. I guess what we are also interested in is to not only know the associations, but also the mechanisms, which is, sort of one layer deeper. So as somebody who's been in this field for decades, what would you say are the most important targets we should start with to try and address mechanisms of all the determinants that we are looking at?
Yeah, I mean, it's a difficult question of how does one do it in a public health epidemiological context, because what's the degree of interventional access to material that you're going to have? That becomes sort of the first question, right? Which is, people have looked at salivary cortisol as a way to start. Salivary and hair cortisol, people have looked at measures that you can actually do, which are peripheral blood work or PBMC dependent. Part of the problem is each of those are fairly interventional because you need to salivary cortisol less so, hair cortisol less so, but still there's still an interventional element where you're going in, you're asking whether, I don't know if you can get necessarily a mechanistic insight as much as a strong correlative insight that might emerge from using more broader tools of epidemiology and public health kind of questionnaire-based approaches where you get a really good sense of the clinical milieu, the situation, the context, and then correlate it with measures that you can actually measure, which would perhaps be reflective of what are the stress levels like, right? I think the interesting thing that will be really interesting to ask is in the socio-cultural context of India, what are the protective factors that play such a critical role despite the adversity and the trauma that may be there, which might be one major factor is poverty and nutritional deprivation, right? Two major factors. And despite that, there will be other factors that overlay and are protective. And that protective factor becomes quite interesting to study because it's going to be highly variant in a socio-cultural context as well. What is it in this context that exerts strong protective effects? Nutrition becomes a very interesting thing to also study in this regard. If one thinks about it in terms of government policies of midday meals / incorporating ways via which to come in with sufficient micronutrient, availability, etc. These kinds of things can be measured because you can measure B12, vitamin D, micronutrient availability. So those kinds of things become things that you could actually measure and then have other sort of hallmark indicators that you can trace over a period of time. But mechanistically, I guess I'm thinking mechanistically in terms of trying to understand what actually happens within the brain. That's going to be difficult to do unless you have an incorporation of an element of fMRI imaging, PET scanning, etc. And that in this early developmental window seems a little unlikely. Hasn't happened extensively world over because of precisely that reason. I don't know. It's not a really good answer, Debarati. I think it's very important work and critical to do. And yet mechanistic insights are sometimes hard to get. What you can do is inspire the work that will perhaps provide the mechanistic insights, because it's this work that drives those more experimental approaches in non-human primates or in rodent models, etc. And then one can toggle back and forth and see what of those measures can be actually addressed from peripheral samples or samples that have minimal level of interventionist requirement in children, adolescents, etc.
Yeah, thank you so much. I had a follow-up question, if I may. So in one part of your talk, you mentioned having identified biomarkers in peripheral samples which indicate early exposure to stress. Is that only in rodent models, or are they also applicable to human models?
We don't know. We'd love to know. I mean, it'd be fantastic to find out. We know in rodent models that we have a decline in circulating IGF-1. So that's serum IGF-1. We know that inflammatory markers are through the roof and fairly early, including CRP. We've not looked at homocysteine, but it's going to be interesting to see how much of that holds true in the human context as well. So peripheral inflammation, markers of peripheral inflammation, markers of sort of peripheral metabolic markers, which seem to show up fairly early in these animal models. Some of them have been looked at in humans. A lot of work has been done in humans with early adversity and telomere shortening in PBMCs in the periphery. That is again another, and that holds true in rodent models as well. So when you do work in rodent models of early adversity, you see telomere shortening. You see the same thing in humans with early adversity.
Mm-hmm. Yeah, thanks so much. Maybe we'll get in touch with you as the study progresses.
Look forward to
thank
interacting.
you.
Absolutely.
Thank you.
Thank you, Debarati, for that wonderful question. It's very fascinating to see how insights from one discipline can inform another. There are two questions from YouTube. I'm going to take them first, and then Supriya has also raised her hand. Okay, so there's one question on YouTube from Jayesh. Are there any changes in cortisol levels observed when GQ receptor is stimulated?
Good question. We have not seen any major change in circulating cortisol or in GR-MR receptors, for example. But we have not looked at it extensively enough to be sure that we are not impacting the pathway. So on the brief first pass answer, I would say doesn't look like it's disrupting it quite dramatically. And that's also true even in maternally separated animals, that just circulating corticosterone in this case, because it's rodents, doesn't give you an immediate sense of the life history. But feedback regulation of that pathway might, and we have not tested that. And that's something we absolutely should do. Yeah.
Okay, I'll take another question from YouTube. Kirti says, as a sociologist and social policy researcher on ACE, I'm wondering on human implications. Parenting and teaching pathway to avert ACE, especially in the context of COVID-19.
I think it's a great question. I'm afraid I don't have the expertise to necessarily answer that at all, but I can tell you that even in animal models, it's fascinating to see the factors that buffer and promote resilience, and many of them are nurture associated and environment associated. And that's been studied quite extensively in animal models, where cross-fostering, adoption, enriched environment, exercise, all of them can have profound effects. And so that tells us that the system, even in relatively genetically identical mouse models and rat models, where the genetic variability is of course much narrower because we're looking at fairly similar strains of, genetically fairly identical genetic strains, even there, environment has a profound effect where you can see reversal, you can see overriding. Nutrition, by the way, is again another key player. Nutritional supplements will become very relevant in this regard as well. So I would be, the implications at the human level where the genetic variability will be substantially greater. I can imagine that the environmental influence on being able to override, promote resilience, and promote supportive players, parenting, grandparenting, neighbors. Essentially, it's a broader community. It's not just the parents in the case of the primate societies, non-human included. The minute you have that complexity of interactions, there are going to be multiple players that are going to promote resilience. And those become very, very interesting to study. At the animal model level, we can definitely model some of these and look at them. Then we do look at effects of exercise, for example, and they're profound on the brain. So they're really quite remarkable.
All right, thank you, Kirti, for that question. Supriya has raised hand. Supriya, you want to go?
Yeah, just thanks, Vidita, for a great lecture. I just had a question on one of your last few slides, which spoke about, investigating ways by which you could potentially reverse the mechanism that you found of the sort of overactive serotonin receptor. And I was just wondering whether that's work that's begun. Do you have any insights on that you could share?
Yeah. So we've done experiments where we blocked the 5-HT2 receptor. And when we did that in at least 2 models, we were able to completely reverse the effects on anxiety and despair-like behavior and on sort of neuronal architectural changes and several gene expression changes. So those were drug blocker experiments. But those are broad. I mean, those were like, systemic administration of the drug. Most of them required early interventions. We needed to go in while the trauma was happening, not later. So that was a big caveat in that that's very unlikely to happen in real life. You want to see things that could potentially reverse the consequences of something that happened earlier. We've done a few studies where we've done pharmacological studies with classical antidepressants like amitriptyline. They reverse the aging-evoked epigenetic changes. They reverse the aging-evoked cognitive decline that's seen in animals with early trauma. So there's a bunch of things that did get reversed, but a bunch of things that didn't get reversed. So it's a partial story there. What we are now trying to do is see if we can actually go in and tweak select circuits using these chemogenetic tools, because we now have a reason to believe that it is forebrain excitatory neurons. In the beginning, we didn't know where it was in the brain. So now it's at least that narrow, even though it's still fairly broad. We need to narrow it even further. We believe it's excitatory neurons of the forebrain that drive at least a subcomponent of the behavioral phenotype. Not all of it, but a subcomponent. So now we want to see if we can modulate the excitation inhibition balance in those neurons in animals that have had a normal history or in animals that have gone through trauma. And if we can do that both while the trauma is going on or later, much later, long after the trauma is gone, and see if we can reverse that. So those are experiments underway. What I will tell you is that while enhancing excitation in these forebrain excitatory neurons by driving GQ signaling mimics what happens with traumatic environment, the reverse, which is to reduce excitation, doesn't seem to do anything, right? So we've done those experiments, not published yet, doesn't seem to do anything. Now, we have not done that in the backdrop of trauma, and that's the sort of next series of experiments we need to do. They're slightly tricky experiments because it involves lots of bigenic mouse model crossing and you need large numbers of animals, but we'll get there. I'm most interested in those strategies that come in later. So I'm equally interested in the exercise / caloric restriction / nutritional supplement strategies, even more so sometimes in the pharmacological, and because I think they might have implications for intervention that are more real world like. And so we want to look at those.
Thank you, Supriya. Anand Bhan has raised his hand. Over to you, Anand.
Thank you, Salik, and thank you also, Vidita. That was an absolutely riveting talk and very, very interesting work that you and your lab and all the collaborators have been doing. I was just curious for Sangath as an organization, which, as you were rightly pointing out, does a lot of work on the ground primarily, but also has a large team now working on early child development. What are your thoughts as someone who's been looking at this issue in the lab for a long time? What are the kind of streams of work you think would be useful for field-based teams to explore based on the insights that you're gathering? And I was also curious, unfortunately, we are in the pandemic. So I was also wondering if you have any thoughts around the kind of stress, etc., that you describe, and that probably also predicting some kind of adverse outcomes. Do you see the ongoing pandemic and the stress it has brought on families, including nutritional deprivation, economic deprivation, the fear of the pandemic, etc., also hence playing a role in terms of impact on future generations? And is that something policy-wise we should be looking at based on your work in the lab as well?
Thanks, Anand, for those questions. I think let me take the second one first, which is I think we're at the, because there's been so much focus on survival associated with the COVID pandemic, we haven't yet tapped the other consequences and the fallouts of this pandemic that are going to be so broad, including on mental health. I think we're just beginning to get a sense of the scale of what we are going to be sitting on as far as that is concerned. In animal models, social isolation is one of the biggest stressors. And, it is a very, very strong stress. And that's an element that one has seen repeatedly in animal model studies. And of course, with species like ours, we're very social species, a social isolation element of COVID. I'm not even considering currently the economic burden that is going to be substantial, and overlay that with the social stress, overlay that with the collapse of support structures, which one could draw on in the past and which now have been overstretched and overstrained and are really now, you're struggling to find the support structures that are required. When you overlay these multiple hits on top of each other, we are going to, I think, see significant increase in mental health crises that come in the decade that is going to follow. And that is, I don't doubt that one is going to see that. And it's going to be disproportionate based on the privileges people carry. And that is obviously going to overlay substantially there as well. So I think there's going to be critical need for intervention at the public health level, for health workers, for just concerted effort to look into this matter, because you get this wrong in this window, or you get this piece, then the consequences aren't during only the transitory period of the virus or the pandemic, which presumably, hopefully 4 or 5 years later, one is talking about having subsided relatively. The consequences will be seen decades out as a consequence of this period of time. So I think interventional strategies that factor this in, which look at it at multiple levels, are so critical. I mean, for example, just the educational fallout for children in the 8th grade onwards to 12th grade pool, adolescent, pre-adolescent, adolescent pool, and the consequences that are going to emerge for them. The nutritional consequences that are going to come because of lack of access to job opportunities, and homes losing primary caregivers, primary responsible people losing either loss of life or loss of job or loss of opportunity, and what that will mean for the whole family unit as an entire whole is going to be substantial. And I think the kind of work you all do is going to be very critical to inform people about, especially governments, to think about where investment will be critical, and, the importance of providing, the pillars, right? Primary healthcare and education and nutrition, these three, if they don't become a very strong focus area going forward for the next decade, we'll be paying the costs of this for decades and decades to come. So I think the work you all do is so critical and so important to inform policy decisions appropriately, because the sort of consequences of this to sharpen the differences of privilege is just going to become, it's only going to compound the problems that are already very steep in our country. It's just going to make it much worse and much steeper unless there is a concerted effort to intervene and tackle and actually make it a priority to focus on. I don't see how one can prevent this from actually just widening and sharpening those boundaries even more than they already are. So that's that's unfortunately something I think everybody will have to worry about. You guys, the kind of work you do is so critical and so important at the best of times. This is really the worst of times, and it only highlights the importance of bringing this information back to policymakers, governments, etc., to really make change happen. The first question you asked, which was the question of what would be some of the measurable things that would be interesting. I'm wondering how a lot of this literature sometimes exists without the socio-cultural nuance of it. And because we are looking at literature that's coming from countries other than our own, I mean, there is literature from India as well, of course, but sometimes that, translation of information across countries and across situations doesn't happen very smoothly. And to looking at buffering/resilience factors in our own sociocultural context, especially when overlaid with poverty, becomes extremely important to see what is it that actually helps to promote resilience and reduce vulnerability. And is there a way to strengthen the practices that allow that to happen? That I think is a unique piece of work that could be immensely useful. It will vary from state to state, will vary from obviously economic context to economic context. But I think having that information that one has, and if there is a way to measure, for example, salivary cortisol, hair cortisol, if there's a way to get peripheral blood monocytes, and one can actually look at PBMCs and look at whether there are epigenetic marks, and one can see reversal of some of these, whether it's possible to measure circulating factors which are peripheral inflammation associated, which seems to be a driver of some of these changes in the brain, and to see if there are factors that can override and help to at least attenuate that, would be immensely interesting and important in the long run.
Great, thank you, Vidita. That's very again very useful in terms of sort of thinking through some of these issues. I did have a couple of quick follow-up questions, sort of not directly related to the earlier questions, but also from a socio-cultural perspective, at a young age for children specifically, there is a role for a nurturing environment which communities provide. Now, of course, there are many factors, including violence within families, patriarchy, etc. But the fact that it's not just from probably the mother or even the father who is doing the nurturing, but a larger socio-cultural environment, and of course that can be perhaps a plus in some cases and a negative in other cases. But is that something which at the lab level also is something that you think about, that is there a role for the collective rather than just the mother as an individual? And also linked to that, a lot of your work, as you were describing, of course, involved the mother and the offspring, but what about the paternal role, and has there been research on that at the lab level, and what does that tell us about how it might play out in our community?
There's been a lot of research in the species that I've talked about, where it's looked at paternal transmission at the level of epigenetic transmission from sperm, etc. So paternal stress resulting in epigenetic marks on the sperm, which may have consequences on the fetus. It's a fair bit of work from Tracy Bale's lab, many others that's looked at that. Unfortunately, the species that have predominantly been of use in the community are not biparental species, and they are in laboratory settings in which the care is literally, the caregiver is the dam, and it's a very short-lived caregiver period as compared to other species. Species that have been used less but have involved biparental care, there are strong effects of paternal separation, paternal stress, etc. as well. So those species, just less work done on those. There just is less work. There's a lot of work done in primates, where it doesn't even work with, it's pure isolation. So then you have multiple components of caregiving, and it's absolutely not just the dam or the mother, any of that. It's much broader, and it's a community. In the model systems we use, a sort of a burrow system might be a better approximation, and we don't use that so much, and nobody in the community does. There's just very few sets of labs that actually do that. There's a very powerful tool because it's a more naturalistic setting than the laboratory cage-like setting which we use, which is fairly artificial in some ways. And that is something that is a factor we always have to keep in mind. But Anand, that's a great question, and it's not been looked at sufficiently. People use voles. Voles are, again, depending, especially the species of vole that you're using, again, biparental care, and then you have a lot of influence from both caregivers. Also, there's a community of sort of nursing care provided by siblings, etc. And so there is that element as well. Those species, you can see that it's a much more complex quality of care, much more complex than the kind of models we use, which are fairly limited in what you can actually address. So that's a caveat for the models that are used quite a bit, which is laboratory rats and mice. These species don't have the complexity of care that we see in primates, and certainly not even in other rodent species.
Great, thank you.
Thank you, Anand. We have one comment and question in the chat box. Sheetal says, thank you, Dr. Vidita, for such brain-stimulating session. I'm very interested to know what would be the public health implication of your work and or how these findings can be used to develop public health interventions.
Yeah, I think our work is a bit removed still, in that we don't know how much of what we do can be directly extrapolated, right? So what we do know is that, similar to what is known in humans, stress in rodent models in that early developmental window has very long-lasting consequences, often long after cessation of that stress, that transient early stress. And so that tells us that early stress has a disproportionately longer impact than adult onset stress or adult onset trauma does. So relatively, if you were thinking about it from a public health perspective, it means that paying attention to stressors or stressful events or trauma in the first 15 years of life can have longer term consequences, and you may even be able to ameliorate things across the lifespan if you tackle it in that window, than an adult onset stress, which more often than not tends to be short-lived, and more often than not people manage to buffer reasonably well, right? So that's what it tells us. It tells us that you need to pay greater attention to early stress and early adversity as a risk factor than to adult stress and adult adversity. And that then tells you that that early developmental window in terms of getting healthcare, nutrition, right, is so critical. I mean, if you think about a country like India with a population with a pyramid where you have a very large young population, and then you want to now look at sort of the pillars that should matter: nutrition, education, healthcare. If you don't do those right, you can invest in many other things and you can do many other things, but you're not going to see real traction at the level of your human capital if you don't focus on those areas. Those are absolute building blocks of government, and government must do that. So those are the things that we as a voting public must hold our governments accountable for. And we have to remember that, because those are the pillars. I mean, they are absolutely where one should be investing. And if one is not investing, one is just going to pay consequences for that for decades and decades to come. And so it's nutrition, healthcare, education, those three. I would say those three are the absolute three essential pillars. So public health care, which we've seen the consequence of not investing in. Don't invest well enough in public health care. You can invest as much as you want in defense. You're utterly vulnerable. Within one shot, you can see the entire system collapse if you don't do it right. And so unless we sometimes have short-term memories, which is we remember it for as long as it lasts. And we don't remember it when it comes down to as people holding our elected representatives to account, which is that you must invest in healthcare and it must be done right. And you must invest in education and that must be done right. And that is the responsibility of the government. And we cannot have government exit nutrition, education, and healthcare and put that out to private players. That's just not long-term viable, because then you will disproportionately provide those resources only at one end of the economic scale and not across the entire lot. So I'd say, and then there's nothing new I'm saying here. Everybody's been saying this forever. It's just that sometimes we are not able to hold our systems accountable, and we must find ways to do that more and more.
Thank you, Sheetal. I had a question, and then so we are running close to time, close to the end of the time. Vidita, can I have a question if you allow?
Sure. Of course, absolutely.
Um, you mentioned about maternal separation and then observing the organism, I believe, at a 3-month interval when they are at the stage of adulthood. I was just curious, what about when they are, say, at the later stage of their life, when these organisms are old? What would maternal separation or early adversity have impact at a later stage in life? So
That's a
yeah.
great question, Salik, and that's exactly what we're doing extensively in the lab currently. In fact, our PVF grant funds exactly that kind of work. We published a few years ago that there is this biphasic nature of effects where animals that went through maternal separation transiently in adulthood actually showed strong adaptive effects. They did better on stress associated learning tasks. They actually showed this boost of adaptation and an attempt of the system to kind of buffer, right? So when that was a little surprising, wasn't necessarily what we had predicted. It was very interesting, but it's like this transient adaptation that you see. And then these animals age. And as they age, you can think of aging as a second stress in some ways, because it's the natural process of cumulative stress. And then when you look at those animals, the aging induced consequences are far worse in animals that have had early trauma. So they show steep cognitive decline on a diverse array of cognitive tasks. You see a steep reduction, you see a great increase in inflammation. You see, in fact, some of these models have been used in Alzheimer's disease and neurodegeneration like models to show worsening of outcomes for neurodegeneration. So actually, Salik, the data strongly indicates that these animals age faster. So there's an accelerated aging associated phenotype in these animals. And we are very interested in asking, how late can we intervene to change that? We've done intervention strategies at about 9 months to 8 to 9 months. And when we do those interventions, we can reverse some of these changes. So that's still fairly late, but we're very interested in asking how can we intervene and change the aging associated outcomes. Lots of experiments underway. I hope I'll be able to share them with you. Some of these are things that we're currently doing in the lab.
Sure. Looking forward to it. So there are no more questions in the chat box, and I see no questions on YouTube either. So I think this is a good point to end our seminar. So on behalf of the entire Sangath team, big thank you to you, Professor Vaidya, for taking time out and accepting our invitation. I thank all the participants for joining in this session. And I also acknowledge and thank my colleague Babita and Pooja for all their assistance and support in making this seminar happen. And so with that, I'll just say what I said again, that this seminar will be shared on our social media handles and it will remain in the public domain. So we can end it. I wish you all the very best. Thank you so much for joining. Goodbye and have a good day.
Thank you.